an article with Raphael Levy and two co-authors. She recently published an article in the BMG calling for the retraction of a Lancet article. Last week, she was interviewed as an expert in the ongoing trial of Paolo Macchiarini. Professor Murray, we are looking forward to your talk.
Okay, well, thanks very much for the introduction. So what I'm going to do in my talk is to start by just giving a very brief overview of stem cells and what they are. And then I'm going to tell you about a couple of landmark events in the field of stem cell research that in some way paved the way for the trachea transplant scandal. And then I'm going to talk about the very first trachea transplant that was conducted in 2008. And then we'll look at the consequences for patients and then why it's important to correct the scientific record in order to prevent patients from being harmed. So to start with then, what are stem cells? So these are unspecialized cells and they have two important properties. Firstly, they have the ability to self-renew to give rise to identical daughter stem cells. And they also have the ability to differentiate to give rise to specialized cells. Now, there are different types of stem cells. So we have pluripotent stem cells, such as the stem cells that are found in the early embryo. So this is an embryo just before it implants into the uterus. And in this area of the embryo here, we have the embryonic stem cells. And these cells give rise to all of the different cell types in the body. And then we also have adult or somatic stem cells, which have a more restricted potential. So a good example of an adult stem cell would be the blood stem cells or hematopoietic stem cells. So these cells are found in bone marrow and also in umbilical cord blood and in peripheral blood. And these give rise to all of the different cell types in the blood, including red blood cells, all the different types of immune cells and platelets. So the properties of pluripotent and adult stem cells are quite different. So as I've mentioned, the pluripotent stem cells can give rise to any cell type in the body, whereas the adult stem cells have a more restricted potential. The first mouse embryonic stem cell line was isolated in the 1980s, and the researchers responsible for that were awarded the Nobel Prize in Physiology and Medicine. So one of those researchers was Martin Evans from the UK. But it wasn't until 1998 that the first human embryonic stem cell line was isolated. And this was by an American group led by Jamie Thompson. And this was a real landmark event in the history of regenerative medicine research and gave rise to a great deal of excitement in the field because we know these cells can give rise to any cell type in the body. So there was a lot of optimism that these cells could be used to treat a variety of degenerative diseases, such as things like Parkinson's disease, age-related macular degeneration, and type 1 diabetes. But the excitable press coverage at the time led to unrealistic expectations, and patients and the wider public were led to believe that these therapies were just around the corner and could be available in just 5 years or so. But it's actually taken more than 20 years to get to the point where these cells are now starting to be used in clinical trials. And that's because it's taken a long period of time to develop the conditions to drive these embryonic stem cells to the required cell types. Now, one of the issues with human embryonic stem cells is that the isolation led to global ethical debate. And that's because the isolation of these cells requires the embryo to be destroyed. So when we're isolating embryonic stem cell lines, we have to remove these outer cells from the embryo and then take these inner cells, which contains the embryonic stem cells, and plate them into a culture dish. So some people feel uncomfortable about the fact that the procurement of human embryonic stem cell lines requires human embryos to be destroyed. And in some countries, it's not allowed to isolate these lines. So in Europe, Austria, Germany, and Italy, have quite a restrictive policy, and it's not possible to do that there. And at the time in the US, George W. Bush banned the use of federal funds for human embryonic stem cell research. Things are more permissive now in the US, but not so in some European countries. And this brings us to the second landmark event, which happened in 2002. And this was a paper published in Nature by a group led by Catherine Verfay, who was in Minnesota at the time, but now works in Belgium. And the title of this paper was Pluripotency of Mesenchymal Stem Cells Derived from Adult Marrow. And what this paper was claiming was that cells found in bone marrow had the similar properties to embryonic stem cells and could generate a wide variety of cell types in the body. However, this research could not be reproduced, and later data discrepancies were identified. But unfortunately, this paper still hasn't been retracted, and it's now been cited more than 4,000 times. And there's some information about this on the For Better Science blog that's hosted by Leonard Schneider. And Elizabeth Bick, who many of you might have heard of, has critiqued this paper and found some problems with the data. And then after these cells were found in bone marrow, mesenchymal stem cells were then found in various other tissues, such as adipose tissue, umbilical cord tissue, and dental tooth pulp, etc. So what are mesenchymal stem cells? Well, these cells were first isolated by a researcher called Friedenstein in 1974. So they're described as fibroblastoid, and that's largely because of their spindle shape, and they have a high growth potential. Now, the reason Friedenstein was interested in these cells was because he was trying to develop conditions in the laboratory that would enable those blood stem cells I mentioned to be cultured. And in the bone marrow, it appears that these cells are important for the maintenance of the hematopoietic stem cell population. So that was really why they first started to be isolated. But then when other researchers started to work on these cells, there was some evidence to suggest that they could differentiate in the laboratory into cells that had some of the characteristics of bone cells, we call those osteoblasts, cartilage cells, chondrocytes, and adipocyte-like cells, so fat cells. But importantly, when these mesenchymal stem cells, as they were then called, were implanted in vivo, typically in animal models at this point, there was no good evidence that they could generate these cell types in an animal. There were also quite a number of other papers around this time that were suggesting that mesenchymal stem cells could generate various other cell types such as neurons, cardiomyocytes, etc. But there is really no robust evidence to support that. So one of the problems with the mesenchymal stem cells is that because of this claim that they were pluripotent, it gave rise to quite a lot of hype in the field and unfortunately opened the door for unscrupulous practitioners to use these cells in unproven therapies. But I'm not going to talk so much about those aspects today. And it's important to point out that at the moment, there's only two widely tested stem cell therapies that are known to be effective. The first is bone marrow transplants. So you've probably heard about people having a bone marrow transplant when they have leukemia. And the active ingredient in the bone marrow transplant are those blood stem cells or hematopoietic stem cells. So the way this works is that the patient with leukemia would have chemotherapy to kill off their own cancerous blood stem cells. And then they would be injected with a donor bone marrow. And the hope is that the stem cells in the donor bone marrow would then engraft in the patient, and then the patient would become cancer-free. And the other routinely used stem cell therapy is in skin grafting. So when a patient has a burn, then they will typically have skin grafted from another part of the body, and the skin stem cells in that graft will then repair the burned area. But these problematic papers about so-called mesenchymal stem cells, which made a number of false claims, which included the fact that stem cells can be harvested from any organ, including the heart. We now know that that is not correct. And there was also the false claim that stem cells can directly replace damaged tissue. And we also know that in the case of mesenchymal stem cells, that also isn't correct. And more recently, the scientific community has woken up to the problems faced by stem cell hype, particularly in the area of mesenchymal stem cells. And a researcher called Arnold Kaplan, who was the first to coin the term mesenchymal stem cells, published a paper in 2017 asking for the name of these cells to be changed. And those of us working with these cells now prefer to call them mesenchymal stromal cells, which underscores the fact that these cells are not really stem cells. And then in 2018, there was a paper in Nature, again, drawing attention to the problems with mesenchymal stromal cells in particular. And then in 2019, the FDA has become more proactive in warning patients about these cells and trying to clamp down on unscrupulous stem cell clinics. So this brings us to the first report of a trachea transplant, and this was published in The Lancet in 2008. And the key players were Italian surgeon Paolo Macchiarini, and he performed the surgery in Barcelona, and two UK researchers who were both based in Bristol, Martin Birchall, who was also a surgeon, and Anthony Hollander, who is a basic scientist. So the patient who received the first transplant was a woman initially from Colombia who was living in Spain called Claudia Castillo. And this is a headline from the New Scientist in 2008, which describes this as a windpipe built from her stem cells. And the University of Bristol in 2010 announced that, this stem cell breakthrough was to enter a permanent national exhibition in the museum. And what they say here is groundbreaking work by researchers at the University of Bristol that signaled a new age in surgical care by helping to save a woman's life. And that's been celebrated in the Science Museum's new exhibition. So I'll come back to this a bit later in my talk to see, well, was it really the case that this intervention saved this woman's life? So what was the clinical indication of this patient that led the researchers involved to try this new approach? So the main problem of Claudia was that she had a stenosis of her left main bronchus, and that was secondary to TB or tuberculosis. So you can see that here in this image. So the arrow is pointing to the area of the bronchus that's stenosed or has a narrowing. So Claudia was told by Macchiarini, who was looking after her at the time, that her only treatment option was either to try this new approach or to have her left bronchus and lung removed. So that's called pneumonectomy. So the proposed strategy was to replace that damaged section of Claudia's windpipe with a decellularized donor trachea from a deceased donor, and then to seed that with Claudia's own cells, the idea being that that would form a personalized replacement organ for her. And the intervention was performed under compassionate use. And this is quite an important point, because usually when a new type of procedure is being tested on a patient or a new drug, the expectation is that it's first tested in the laboratory, then on animals, and only if it's found to be safe and effective in animals is it then tested in humans, and usually in a clinical trial that has regulatory and ethical approval. But there are some circumstances when new interventions can be tested outside a clinical trial if the intention is to try and save a patient's life. And in this situation, a doctor can try a new treatment, but the important point is that the doctor has to have the best interests of the patients at heart. And whatever the new intervention is that's being tested, there has to be a very good scientific rationale that, underpins why the doctor thinks it's going to be helpful. So what happened in this first trachea transplant? So this was the strategy. So first, a donor trachea from a deceased donor was decellularized. And this was done by exposing the trachea to detergents and enzymes. And typically about 25 cycles of this type of treatment is required in order to completely decellularize the trachea. The next step was to take the mesenchymal stem cells, as they were then called, from Claudia's bone marrow, and then to direct them to become chondrocyte-like cells in the laboratory. And those chondrocyte-like cells were then seeded onto the outside of Claudia's donor trachea. Now, the idea was that these chondrocytes would give the airway some structural support so that it wouldn't collapse following implantation. And then the next step was to take some of Claudia's bronchial epithelial cells, grow them up in culture, and then seed them onto the inner surface of the airway. And it's very important for the normal functioning of an airway that the airway has a lining of these cells, because these cells are very specialized. They're ciliated, and that's important to clear mucus from the airway and to help prevent colonization of microorganisms such as bacteria and fungus. And then after the cells had been seeded on, the trachea was cultured in a bioreactor for 4 days. And this is an image here of the bioreactor growing in the laboratory. And this is the patient's outcomes as reported in that Lancet paper. So this is the image before the transplant. And this is an image taken 1 month after the transplant. So you can see now that there is no stenosis here in the bronchus. And this is a quote from the abstract of the paper. So it says, the graft immediately provided the recipient with a functional airway, improved her quality of life, and had a normal appearance and mechanical properties at 4 months. And also immunosuppressants were not required because all of the cells of the original donor had been removed through those detergent and enzyme wash steps. But the paper did lead to a number of questions. So the first question was, had the patient's cells really integrated into the donor airway? And the researchers themselves were not able to answer that question. And in the discussion of the paper, they do indicate that there was actually no proof that the cells that were seeded on did anything. Another question was, how did the trachea that was implanted stay healthy without a blood supply? And this was a question that was raised immediately by a surgeon in Leuven called Pierre de Lara. And he wrote a letter to the Lancet, which was published, raising concerns about this issue. Another question was, did the trachea maintain its structural integrity? And this was a question raised by a Chinese researcher who also wrote to the Lancet and was pointing out that it seemed extraordinary that, seeding these chondrocytes on would give the trachea sufficient structural integrity that would keep it open or patent. And one of the very surprising things about the paper was that Claudia didn't have a stent placed in her implanted trachea to keep it open. And there was no reports in the paper that, the trachea had any problems. Another question was, had the approach been tried in animals? Was compassionate use justified? So was this patient's life really in danger? And was the patient aware of the potential risks of the intervention? And I'll come back to these questions a bit later. And then another question was, had the necessary regulatory and ethics approvals been obtained? So after Claudia had her bioengineered trachea implanted, and it's important to note that with Claudia's operation, the trachea was used to replace one of her bronchi, not her trachea. But afterwards, 11 more patients were treated, and in most cases, the subsequent patients had their trachea replaced. And you can see information about all of those patients on Leonard Schneider's For Better Science blog. And Leonard has really done a lot of work in uncovering, some of the problems with trachea transplants. It's also important to point out that the strategy did change with subsequent patients. So the next patient who was operated on in Barcelona after Claudia didn't have her cells seeded on a decellularized airway and incubated in a bioreactor for 4 days. Instead, she had her cells seeded onto the trachea at the time of the transplant. So, although Macchiarini is the person who really pioneered this, this intervention, Martin Birchall also continued with some additional patients in the UK that didn't always involve Macchiarini. So this is a screenshot from the website of University College London in 2018. So after the first transplant in 2008, Martin Birchall relocated to University College London. And here they have images of 3 patients who had these type of airway transplants: Claudia Castillo, and then Kieran Finn Lynch was the first child to have a trachea transplant. That occurred in 2010 and was published in The Lancet in 2012. And another patient called Shauna Davison was implanted in 2012. So Macchiarini wasn't involved with Shauna Davison's transplant. And this is a quote from this webpage, and it says, "Our progress has been via life-saving breakthroughs in tissue engineering technology in one-off compassionate patients." And as we see here, because of the alleged success of these transplants, University College London were planning to proceed to clinical trials, which would involve more patients getting these type of trachea transplants. So from Barcelona, Macchiarini first relocated to Florence in Italy, where he worked in the Correggi Hospital and undertook 5 more transplants with cadaveric donor tracheas. And after that, he was headhunted by the Karolinska Institute. So that was towards the end of 2010. Now, in the Karolinska, Macchiarini switched to a different approach for trachea transplants. And instead of using decellularized donor tracheas, he switched to bioprinted plastic tracheas. And the reason he gave for that was because it's not very easy sometimes to find a suitable donor. And also there were some problems with the decellularized tracheas collapsing following implantation. So the first patient to receive a plastic trachea is this patient here called Andemariam Bayen. And that procedure was performed in the Karolinska University Hospital in 2011. And it was reported in the Lancet just a few months later. And it was described as a stem cell seeded bioartificial nanocomposite. So what was done in this procedure was that a plastic trachea was made for Andemariam, and then cells from his bone marrow were aspirated and seeded onto the plastic trachea a couple of days before the plastic trachea was implanted into him. And the idea was that, as the title of the paper suggests, was that those so-called stem cells in Andemariam's bone marrow would transform the plastic trachea into a functional organ. Now, the problem with this approach is that the only bona fide stem cells that are found in bone marrow are the hematopoietic stem cells, those blood stem cells. And we know that they're not cells that would be able to generate a trachea because they're cells that give rise to different types of blood cells. In total, 9 patients have been implanted with the plastic tracheas. In 2014, 4 doctors at the Karolinska University Hospital raised concerns about these trachea transplant operations because the patients were experiencing very serious adverse effects, and these interventions were causing severe suffering to the patients. So this is an image of the 4 whistleblowers here, and this is taken from the For Better Science blog. And they submitted a very detailed report about the patient's adverse effects to the people at the head of the Karolinska, but their concerns were brushed under the carpet. And instead, they themselves were threatened because it was alleged that they'd breached patient confidentiality by raising their concerns, which wasn't in fact true. And unfortunately, because of these concerns being brushed under the carpet, it enabled more transplants to take place. And it wasn't until 2016 that that action was eventually taken. And this was as a result of a documentary being published, being broadcast first in Sweden and then in the UK in English. And the English version was called Fatal Experiments, The Downfall of a Supersurgeon. And it consisted of 3-hour-long episodes. And this documentary really highlighted the suffering that these patients experienced, and also highlighted how people at the head of the Karolinska hadn't taken appropriate action and had tried to sweep things under the carpet. And after this documentary was broadcast, the people at the head of the Karolinska had to resign. Around the same time that the documentary was broadcast, an article appeared in Vanity Fair. So this was in February 2016. And this was about Macchiarini's personal life. And it's called "The Celebrity Surgeon Who Used Love, Money and the Pope to Scam an NBC News Producer." So the news producer in question is this woman here called Benita Alexander. This is Macchiarini. So Benita Alexander was making a program about Macchiarini and fell in love with him. Macchiarini proposed marriage. But the problem was he was already married but hadn't revealed those details. And Macchiarini was planning the wedding, and they were meant to be getting married by the Pope, and famous celebrities were meant to be attending the wedding, such as Elton John and Tom Cruise. But of course, this all turned out to be a fantasy. And this was published around the same time. So what about those 9 patients who had the trachea transplants? Well, a very thorough report was published by a BBC journalist called William Kremer. And this is a table from his report. And what we can see here is that of the 9 patients, only 2 were still alive at this point. Unfortunately, Yesem Chater has also since died. She was the 3rd patient to be operated on at the Karolinska. And there's only 1 patient who didn't die, and that was because his synthetic trachea could be removed and he could have a tracheostomy. That wasn't possible for the other patients because the plastic trachea extended into their thoracic cavity. So once that had been done, there was nothing really that could be done to help those patients. In 2019, the whistleblowers published a very detailed report, which is publicly available. And it really goes into detail about what happened to the patients after these plastic tracheas were implanted. So this is an image from their paper showing a normal airway, and those arrows are pointing to small blood vessels in the lining of the airway. And this is what one of the plastic tracheas looked like. So for some of the patients, it wasn't just the trachea that was replaced, but also the first part of their 2 bronchi. And this is an image from the paper showing a bronchoscopy of the first patient at 2.5 months. And those blue sutures that you see there, this is where the plastic is being sutured onto the normal part of the patient's airway. So this is a quote from the paper. So it describes this as follows. So there's an obstruction of the right distal anastomosis. So this is referring to this part here. So this is what a normal airway looks like. And this is what the first patient's one looked like. And also there was a left distal anastomotic fistula, which is shown on this side. Now, the development of fistulas was something that happened in all of the patients at the Karolinska just a few weeks following the transplants. So what happened was the plastic trachea wasn't really able to integrate with the remains of the native airway and would start to separate. And as a result, a small hole, which we call a fistula, developed. And that was really bad news for the patients because it meant that air that they breathed in could leak into the thoracic cavity, and this caused very serious complications. Another common complication that the patients experienced was that the plastic trachea would damage their esophagus, which lies directly behind the trachea, and caused fistulas to develop in the esophagus as well. So that if the patients tried to eat or drink anything, that would leak into their thoracic cavity. So many of these patients weren't able to eat by mouth anymore after this happened and had to be fed through a tube in their stomach. So thanks to the documentary, the Karolinska University Hospital did a very thorough investigation into the 3 trachea transplants that had happened there. And the Swedish Central Ethics Review Board found that Macchiarini was guilty of misconduct and requested the retraction of 6 of his papers. So that first paper that was published in 2011 about the trachea transplants was then retracted. And then at some point after that, Macchiarini was indicted for aggravated assault. So this was back in 2019, but there have been delays for various reasons, including because of COVID. And the trial is actually still ongoing, but is meant to complete next week. And the judges will probably take a few weeks to deliberate and then, decide what the verdict is. So as was mentioned at the beginning, I've been an expert witness in the trial, and that information is in the public domain. But as you'll understand, I'm not able to say anything about, what happened during my appearance at the trial or what evidence I gave. But it's interesting to point out that the prosecutor is indicting Macchiarini for aggravated assault, not manslaughter. And that's really to underscore the appalling suffering that these patients experienced. And the patients who survived for the longest time, suffered the most. And the third patient at Karolinska, Yesem Chater, she was 4.5 years in intensive care after her trachea transplant and could never leave intensive care, and had a total of over 7,600 bronchoscopies, over 100 surgical interventions. So it was really appalling suffering that she had to endure. So after all of this information came out about the plastic tracheas, questions then were asked about, well, what happened with the patients who had those cadaveric tracheas? And was the outcome of that very first patient, Claudia Castillo, accurately reported? So shortly after the scandal broke in Sweden, Leonard Schneider published an article on his blog called Claudia's Trachea. So this was in November 2016. And this was where I initially realized that, yes, there were problems with that first trachea transplant. And there's quite a lot of information about that on Leonard Schneider's blog. So what happened to those 11 patients who received cadaveric transplants after Claudia? Well, we know that 9 are dead. 1 is a survivor, and that's Kieran Lynch, the first child to have a trachea transplant. And 1 patient is unreported. The unreported patient was a child who was age 3 in May 2017 and had a trachea transplant at Great Ormond Street in the UK, but no outcomes have yet been published for that patient. So another question that many people had was, well, what about the planned trials that UCL were involved with? So there were 3 trials altogether. Inspire and Tetra were trachea transplant trials. Inspire was UK based, whereas Tetra was funded by the EU and was going to be a European wide study. And then Regenvox was a larynx replacement trial, which was to take place in the UK. So University College London announced that they were conducting a special inquiry in September 2016 about their role in trachea transplants. And I think the expectation of most people who knew about what had happened to the patients was that they would recommend the trials wouldn't continue. So the results of their inquiry was published in September 2017. But to great surprise, the inquiry recommended that the trial should proceed, even though most patients who'd had this procedure had died. So it was at this point that myself and Raphael wrote a report about the trachea transplants and submitted it to a parliamentary inquiry into research integrity that fortuitously was taking place at the time. And this allowed us to raise our concerns about these trials that were planned to take place. And we were pointing out that, you know, patients who'd had these previously had experienced adverse outcomes, the most common being death. So after this was published on the parliamentary site, the story was then taken up by Science, who published a paper in 2018, again raising concerns about these trials. And then the Telegraph published a story in the UK that was focusing on adverse outcomes of 2 UK patients. And then Newsnight, which is a BBC programme, also did a story about it. So thankfully, the trials didn't go ahead. But what happened to Claudia Castillo, and was that paper, you know, an accurate account of what had happened to her? So just to quickly go through these questions. So had the patient's cells really integrated? Well, there was no evidence that they did. How did the trachea stay healthy without a blood supply? There's no evidence that it did. And we know that the trachea collapsed after just 3 weeks. So it didn't maintain its structural integrity. And after 3 weeks, Claudia had to have a stent inserted into the trachea to keep it patent. And if Claudia had had a trachea transplant rather than a bronchus replaced, then it's quite possible that she would have suffocated and died. But because it was just her bronchus that was affected, she was still able to breathe through her other bronchus. Had the approach been tried in animals, there's no evidence to suggest it had been, although Macchiarini and Burchell claimed that they had tried it in pigs. Was compassionate use justified? So there have been claims that Claudia's life was in danger, but from a freedom of information request I obtained from the hospital in Barcelona where it took place, it's quite clear that Claudia's life was never in danger. So that raised questions about whether it was appropriate to try this unsafe technique. And was the patient aware of the risks that she was taking? Had regulatory and ethics approval been obtained? Well, we know that the regulator in the UK, the MHRA, hadn't given approval for a trachea to be bioengineered in Bristol. And the patient samples, the bone marrow and the epithelial cells, arrived in Bristol before ethics approval had been granted in Spain. So there are some concerns about that. And then I just wanted to quickly mention this animal study that was meant to have taken place prior to Claudia's transplant. So this was published in 2010. And it says here in the paper, we recently performed the world's first stem cell-based, fully tissue-engineered tracheograft, which has been a success to date. However, before we used the graft in human subjects, we evaluated the relative contribution of the autologous biopsy-derived epithelial cells and mesenchymal stem cell-derived chondrocytes for the survival of the scaffolds, and this was all done in pigs. Now, this paper is highly problematic. So the first problem is that the methodology is false because they describe exactly the same procedure for preparing the chondrocytes that was used for Claudia Castillo's intervention. And we know that that can't have happened prior in an animal study because the specific conditions had been invented by Anthony Hollander, who wasn't involved in this pig study. And he'd actually patented that technique, but the patent hadn't been published. So we know for sure that the methodology in the paper had to be false. And that, of course, raises concerns about the whole paper. But the other problem with the paper is that these are the main results. And this is showing the outcomes in those pigs who had the transplants. So these pigs were implanted with decellularized tracheas that didn't have any cells seeded on them. These tracheas were from pigs that had implanted decellularized tracheas with chondrocytes on the outside. These pigs got tracheas with bronchial epithelial cells on the inside, but nothing on the outside. And then these tracheas were from the pigs that had chondrocytes on the outside and epithelial cells on the inside. And the claim is that these are now completely normal tracheas. But this data doesn't support that, because if you want to claim that, the data they should have provided was, instead of a cross-section, a longitudinal section where we can see the join with the native trachea. And the other issue is that this is scientifically implausible, because we know from other studies that if you take a trachea from an animal, such as a rabbit, remove it and immediately sew it back in, it won't be a normal trachea, because there will be some granuloma tissue and there'll be some narrow in there. And what these researchers are expecting us to believe is that instead of taking part of the trachea out of a pig and immediately sewing it back in, if instead we dip the trachea in detergent and enzymes and then seed some cells on it and implant it back in, it will be a completely normal trachea. I mean, and this is just completely implausible. And I have raised concerns with the journal who published that paper and haven't received a response. But of course, it's also important for that very first trachea transplant paper to be retracted. And, you know, many people have really, you know, raised concerns with the Lancet about this, but they refuse to retract it. And, you know, as was mentioned earlier, Raphael and I and two of our colleagues, Leonard Schneider and Peter Wilmshurst, wrote a letter in the BMJ saying that it's really time now for this paper to be retracted, but the Lancet still won't retract it. So why is it important to retract this paper? Well, the most important reason is to prevent any more patients being harmed. And secondly, it's also important to correct the record in order to respect the patients who were harmed by this fraudulent research. And, you know, the reason why it's important to retract is this same strategy is about to be tested in a clinical trial that is due to start in October in the UK, which will use exactly the same approach, take a donor trachea, decellularize it, implant it with bone marrow cells, and then take a patch from that to repair the hole that patients have in their bronchus, sometimes as a result of having their lung removed. And this condition is called bronchopleural fistula. But there is no evidence that this approach works, and actually quite a lot of evidence that it doesn't. But the company who's sponsoring this trial, Viderigen, they applied for funding in the UK and they used that 2008 trachea transplant in their funding proposal. So the big question is, you know, why is it possible that these unsafe procedures continue to be happening? You know, Viderigen have got approval of our regulator, they got funding, they've got ethics approval. How is it possible for them to obtain those approvals without any evidence that the procedure is safe, and actually quite a lot of evidence that it isn't? And I don't know the answer to that question. But I would encourage you to look at Viderigen's website just to see for yourself what they propose. So this is the instrument for decellularizing organs. So the idea is you put your donor organ in there and then enzymes and detergent is applied and you end up with a decellularized organ. And then we see some blood being taken here from a patient, and then the blood is injected into the instrument, and then we have a new organ for the patient. I mean, this is just completely ridiculous. And, you know, some of you may have heard of the Theranos scandal, but I think in comparison to what Theranos proposing, this is actually even more implausible. So to summarize and complete my talk, the message I'd like for you to take home is that bad science can harm patients and waste money. And bad science in regenerative medicine also risks damaging bona fide research that's taking place in this area. And then finally, I just want to acknowledge people who've really helped to, stop the trials in the UK. So that includes Leonard Schneider, who has many stories about this on his blog, Raphael, Peter Wilmshurst, Liz Faulkner, Norman Lamb, who was one of our politicians, and then a number of journalists, Matt Warren, Camilla Turner, and Deborah Cohen. And sorry, I think I've gone over a bit, but I'm happy to take any questions.
Thank you for your presentation. Now it's time for the discussion with Professor Murray. Do you have a question? Firstly, you studied by student and postdoc if you have any question.
Okay, so I can see Didier Latourneau, if I say that correctly, has a question.
Yes.
Okay, thank you. Can you hear me?
Yes.
Okay, so thank you and congratulations for this nice overview of the long story of Macchiarini. I try also to teach a student about this story, but I have the full story with you. It's even better. I have several questions on command. One almost personal question about Macchiarini. What is the situation? What I heard is it's vascular, is a surgeon in Kazakhstan and still doing surgery on per arm, harmful stuff on patient. Do you know what is the situation? Because you said
We have a problem with connection, I think.
Yeah, I mean, I can answer the first question. I don't know what he's doing now. Yeah, so whether he's still doing surgeries, I don't have any information about that, I'm afraid. Can you hear me?
Yes.
Yeah. I think it was Didier who froze. I don't know if anybody else has a question. Maybe he can try and reconnect.
Yep.
Well, Didier is not a student, and I'm not either. So is there any student or postdoc who wants to ask a question?
Yes. Can I? May I?
Yes, please.
Okay, well, first of all, thanks a lot for this presentation and the explanation of this case. Really, really interesting. I have a question. So it's a bit personal also. What was, because I guess there are a lot of cases, well, maybe not like this involving a lot of patients, but there are many cases of fraudulent science. And what was your motivation? And it's really difficult with these people that they are like gods in their fields, and they are publishing in the best journals. So what was your motivation? And also, it's really a risk to do that, because you are exposing yourself also to be criticized and to be punished also by the community. So, well, I think this is very courageous to do this. And what was your personal motivation or professional motivation to do this?
Yeah, so I was interested in the trachea transplants because before going to university, I was a nurse on a head and neck ward. So I was looking after people with tracheostomies, although no trachea transplants were done on that ward. And, you know, I think I was very lucky because, you know, the surgeons were all very good people, and the patients were always, you know, the most important thing, and everything that was done was for the best interests of the patient. And with the trachea transplant story, it really shocked me that, you know, the patients were really being exploited, you know, to help the careers of the researcher. And, you know, that first paper was something that I used to talk to my students about, because I gave lectures on regenerative medicine. And although there was no science behind it, I didn't doubt that a patient had been helped, because I just couldn't have considered that surgeons would lie to that extent. So that was another reason, because, you know, I didn't want to carry on talking about that paper if it was false. So that was why I was also motivated to find out what happened. But then the reason for trying to really do something about it was because they wanted to carry on and do more trials, which I just thought was unbelievable. So I thought it was important to try and stop it, because we could see what the outcome for those patients was likely to be. And in regard to the risks, I think what helped was that, you know, it wasn't our university that was involved in the trials. I think if we'd been employed by University College London, then the outcomes might not have been, you know, so good for us, because I think there would have been a greater risk for sure. Yeah.
Okay.
So
Didier, I think you got.
Yeah, I'm sorry, my computer completely crashed, so I'm back. Sorry for my question. So my first question: what is the situation of Macchiarini? Because if he's still doing this in other countries, it could be really harmful for patients. The second question: if with this story, how can you explain that other clinical trial can be allowed by the story of VD Regen? How the regulatory authority are doing a job which it's probably not correct? That is the situation.
Yeah, so
we know that Macchiarini did the wrong thing. He used a compassionate file, and this is fine. Other companies are doing the regulatory pass, official pass, and they are doing the same thing. So what is wrong also in this? And the last question, it's about the journal. What is the role of Lancet, for instance?
Yeah, so the first question, I don't know what Macchiarini's doing now, but for sure he poses a risk to patients, because we know that the trachea transplant wasn't the only problematic thing he was doing. You know, there were problems with patients with pulmonary hypertension previously, patients who were terminally ill. So I think he would always be a risk to patients. But I don't know what he's doing now. The second question, how is it possible that this company, Viderigen, can obtain ethics and regulatory approval? So I put in a freedom of information request to ask for the copy of their application for a clinical trial. And every page was redacted, you know, every page, even though there is nothing there that is a secret, because they've patented the technology. So why don't the regulator want me to see what's there? I then put a freedom of information request to the ethics committee to ask for the patient information sheet, which again is not a confidential document. And I was told they wouldn't release it to me in case it should jeopardize the commercial interests of Viderigen. So I've since been approaching the hospital, the Papworth Hospital, where the trial is meant to be taking place.
Yes.
And I asked them for the patient information sheet, and they told me the same thing. They won't release it because it would jeopardize the commercial interests of Viderigen. So I appealed to them and I said, well, it would only jeopardize their commercial interests if they're not telling the truth to patients. So the hospital have now released that, and indeed, the patients aren't being told the truth. So we have appealed to the hospital not to allow this trial to continue. Now, it has been delayed because it was meant to be starting last year, and now at least the start is being put back till October this year. So we still have time to try and stop patients being harmed by this unsafe approach. I've also contacted the chief executive of our main funding body in the UK, because Viderigen have been funded to the tune of about £6 million with 3 grants from Innovate UK, which receives government funding. And I said, how is it possible that this company, and we know that the company has included false information about patient outcomes in funding proposals, how can they still be getting funded? And I was just told, well, you know, this can be good for society, this sort of thing. And I think, really? I mean, and this is the chief executive of our main funding body in the UK telling me this. So I think our main hope is that the people in the hospital will see sense and not allow this unsafe trial to go ahead.
Yeah, but there is also a role of regulatory authority because they did allow this.
They did, yeah. And, you know, all I can say is it's not the only problematic case that the regulators have been involved with.
Yeah,
You
but
know,
it's growing, especially in this field.
yes.
In other field also, COVID was plenty of things. Okay.
Yes.
On the last part, the responsibility of the journal, typically Lancet.
Yeah, so why won't the Lancet retract this paper? I mean, we even had one of our politicians, Norman Lamb, writing to them asking why, you know, what's the justification? Because we know it's fraudulent. One thing that could be having an influence is that the chief editor of the Lancet, Richard Horton, he does have an honorary position at UCL. So whether he feels some affiliation there and he feels he doesn't want to retract a paper if it has UCL researchers, but I'm just speculating.
Okay.
I don't really know, you know, it doesn't make any sense.
Okay. Thank you. Thank you very much. Congratulations for your work.
Thanks. Does anybody else have any questions? I mean, I can also recommend an electronic book that's been written by Pierre de Lara. It's called The Biggest Lie in Medical History. And that goes into very, you know, extensive detail about all the cases. And Pierre is a person who right from the beginning has been trying his best to stop these procedures going ahead and has really just been hitting a lot of brick walls, you know, but I really admire him for his tenacity. And, you know, I think he did really have a lot of, you know, influence in the end, even if unfortunately it took some time for people to really pay attention to what he was saying.
Another questions?
No, don't think so.
Okay. So should I stop the recording then? If I can do that.
Is it okay for you?
Okay.